Hi, I'm Daniel. I'm working at the University of Tübingen with the Functional Metabolomics Lab on the development of mass spectrometry tools and their application to study chemical exchange within microbial communities. What I want to show you in this short video is how you can use the GNPS dashboard to quickly visualize extracted ion chromatograms from molecular networks, in particular from the web visualization tools of molecular networks in GNPS. This video is part of the use cases for the GNPS dashboard paper. And for the case study, if we are within a molecular network here in the web browser-based visualization from GNPS, we eventually have a molecular family, for example here Yersinia bactens, in which here the compound with the mass 482 would represent Yersinia bactin, a natural product produced by Yersinia pestis. So now, if we want to quickly visualize this compound, in particular its extracted ion chromatograms, what we can do is we can go to all clustered spectra through this link here, and then we get a list of all MS2 spectra that contributed to this particular cluster. And then here, through this View LCMS chromatogram link, we can directly go to the GNPS dashboard, in which we can specify the mass for the extracted ion chromatogram here, as well as like the precursor tolerance, either in Dalton in absolute or relative in ppm. And when we scroll down, we can see the extracted ion chromatogram of this particular mass here in this window on the right. And what we can see nicely is that it's actually not only one compound, but we have two nicely chromatographically separated peaks, even though in the molecular network, those were only represented as one node, due to the fact that they all have very similar MS-MS spectra. And this is also a very nice example why I think sometimes it makes sense to use feature-based molecular networking instead of classic molecular networking, where you actually first take the chromatographic features into account and then perform molecular networking of the resulting MS2 spectra from these peaks. Fortunately, within the GNPS dashboard, you can also directly perform feature finding onto the file you just loaded in there. So here under the LCMS viewer options, you can actually select different feature finding tools such as MZMine2, and then in this windows here specify some basic parameters such as precursor mass tolerance or MS1 noise level. And then directly by clicking this button here, perform feature finding for the file you visualized before. So now here all picked features are indicated as a green box. And if you're happy with those settings, you can actually go through the Run Feature Finding at GNPS link and basically bring the settings into a ProteoSafe workflow here, where you can select more files and then perform like an MZMine batch processing directly in GNPS. This then also links to actually the feature-based molecular networking workflow from which you can generate your molecular network. And in our case, where I did this already before, I then go to a resulting feature-based molecular network. And here, when we visualize the network and label the nodes by their mass, you see nicely that now indeed we have two nodes for each of the chromatographically resolved peaks. And when we look at their MS2 spectra by left and right clicking on them, we can also see why they were merged before. And again, this is because they have very similar MS2 spectra, because there are some stereoisomers. So now as a next step, we could also visualize then this again in the GNPS dashboard. And here in addition to only one file, I now loaded the strings for a couple of more files, where we actually have a scenario where in the first group we have a knockout mutant, and in the second group we have the wild type. And then by defining here again the mask for the extracted ion chromatogram, we can also directly get the peak areas for actually like both peaks from the different samples. And then by the grouping, we can also display them in the actually average values in a box plot of like the two groups, where we can see nicely that here for wild type we have actually like quite some abundance, and here for the knockout, where the GNPS biosynthetic gene was depleted, we don't have any Yersinia bacteria production. So I hope with this I could show you how you could use the GNPS dashboard for your research, in particular natural product research, but also many other topics, and I hope this is useful for you. Thanks for watching, and see you soon.